Looking at the cell cycle, we know that interphase is 90% of the cell's time in the cell cycle. So this is what interphase looks like. We're able to see chromatid, and it looks very uncondensed. Here's our Golgi apparatus. Remember, with our rough endoplasmic reticulum, smooth mitochondria. Here are the centrioles, which, remember, these are going to appear during mitosis, and these little lines are going to be our microtubules. With prophase, in most books, prophase is split up to early and late prophase. First thing that we're going to notice is that DNA starts to condense. This was our previous cell. Now we're seeing DNA condense. In our original cell, our centrioles were really close together. In our new cell, notice how the centrioles start to move apart. So we are seeing DNA starting to condense, but we're also seeing the centrioles start to spread apart. Remember, the centrioles' purpose is to attach to our chromosome and pull them to opposite sides of the cell. So they're starting to get in position here. The next one we're going to take a look at, this is going to be late prophase. What's really happening here is we're seeing this nuclear envelope start to disappear. We're seeing the DNA has condensed down tremendously from before and definitely from our original. Centrioles are starting to move further away in opposite poles of the cell, and now we have our microtubules that have extended, and they're going to latch on to the center of the chromosomes. There's a structure right here called kinetochore that you can see that the microtubules are going to attach to. This is where we're going to have early metaphase. Our centrioles are on opposite sides, and our kinetochores are attached to these microtubules. Notice how they're getting ready to assemble in the middle, which brings us to metaphase. With metaphase, they are completely lined up in the middle. Metaphase, middle. Our nuclear envelope is completely disappeared. Here we have anaphase. Anaphase, remember, away. Our pairs of chromosomes that we had, so this was metaphase. Okay, remember how we have two sister chromatids making this X? Now we are pulling these chromosomes apart. Our microtubules are shrinking back into the centrioles, and that's what's pulling these chromosomes apart. So eventually they're going to pull away like this. This brings us to telophase or telophase. Anytime you see this cleavage furrow, telophase, I think T like titties because cleavage, y'all get it? Anytime you see this cleavage furrow right here, this is good indication that we're in telophase. We start to see the nuclear envelope start to reappear slightly, which we did not see in anaphase. Finally, we're getting ready for cytokinesis. Our nuclear envelope has reappeared. Our DNA is getting ready to start decondensing, which means we're going to see it look a lot like this. And then eventually it'll be fully decondensed and it'll look like this. Once it starts decondensing, this is where we're going to have cytokinesis, which is where the cytoplasm actually divides. And if you can see, there's a groove here to let us know the cytoplasm is getting ready to divide. Our microtubules have shrunk back down into or around our centrioles, and we can see the rest of the organelles start to reappear.